Okay, so welcome back to this next video in which we are discussing cholesterol metabolism. Okay, so now in this video, what I want to discuss is why hypercholesterolemia, which is too high LDL level within the blood, can actually lead to cardiovascular disease, so strokes and heart attacks, okay? And the way that it causes this is through causing atherosclerosis, or rather accelerating atherosclerosis. Okay, right, so in this video then the key topic that we're going to discuss is atherosclerosis. Okay, now the first thing then that we need to discuss in order to be able to understand what happens in atherosclerosis is the histology of the vessel wall of arteries, and generally atherosclerosis does occur in arteries rather than veins. Okay, so I want to start by discussing the histology of the arterial wall, okay, so that we know the different portions and then we can understand the pathogenesis of atherosclerosis. Okay, right, so I'm now going to draw a picture then of the structure of an arterial wall, and we're going to start from the inside and then work our way outwards. Okay, so the innermost layer then of the arterial wall is going to be the endothelial cells, okay, and the endothelial cells will be sat on a basement membrane. So let me start by drawing the endothelial cells, which are lining the lumen here. So here they are with their nuclei. Okay, I'll just speed up this process a little bit. So here are our endothelial cells, and all of these endothelial cells are going to be sat on a basement membrane, which I'll put in a moment. Okay, so there are the endothelial cells, and then underneath the endothelial cells, what you're then going to have is the basement membrane, okay, which is going to be represented by this turquoise line here. Okay, and the basement membrane is a layer of proteins, basically. It's a protein meshwork on top of which all of the endothelial cells are sat, okay? The reason that the endothelial cells don't just fall into the lumen of the blood vessel, okay, is because they are attached very powerfully to this basement membrane, which is this protein meshwork underneath them. Okay, so let's label up these two components that I've drawn so far. So this is the basement membrane here in turquoise, okay, which will be abbreviating down to BM from now on. So B for basement and then M for membrane. So I'll just colour code this up. So this is the basement membrane in turquoise here. Then the cells that are sitting on the basement membrane here, these are the endothelial cells, which I will just abbreviate down to ECs. Okay, right. So what then do you have underneath the basement membrane? Well, underneath the basement membrane you then have a layer of connective tissue, okay, which I will colour in orange here. And this is going to be the absolutely crucial layer in which atherosclerosis is actually going to occur, basically. Okay, so this layer of connective tissue underneath the basement membrane, this is what's known as the subendothelial connective tissue, or just sometimes called the subendothelial space. Okay, so either the subendothelial connective tissue, or we'll use subendothelial space. Okay, so that's represented in orange here. Okay, now outside then of the subendothelial space, what you're then going to have is a layer of elastic connective tissue. Okay, so the connective tissue in the subendothelial space was mainly collagen, okay, non-elastic connective tissue. Now you're going to have a layer of elastic connective tissue, okay, which I'll represent in blue here, outside of the subendothelial space. Okay, and this layer is now called the internal elastic lamina here. Okay, so in blue, this is the internal elastic lamina, and there is going to be also an external elastic lamina, which we'll see later on. Okay, right, so all of the layers that we've now seen, okay, the endothelial cells sitting on their basement membrane, the subendothelial space underneath the basement membrane, and the internal elastic lamina, all of those layers collectively together, they are known as the Tunica Intima. So Tunica means layer, okay, Intima means close, Okay, so then a layer that is close to the lumen of the blood vessel. Okay, and I should have probably pointed out the lumen of the blood vessel. That's that portion in there. Okay, so all of this is the Tunica Intima, basically. Okay, and atherosclerosis is primarily a disease of the Tunica Intima. Specifically, it is going to occur in the subendothelial space, but it occurs because of the endothelial cells. The endothelial cells are the principal drivers of atherosclerosis. They're going to be the ones that are misbehaving, basically, and causing all the problems. Okay, right. Then, outside of Tunica Intima, or more immediately outside of the internal elastic lamina, you're going to have a layer of smooth muscle cells, okay, which I'll denote in red here. Okay, so this is many layers of smooth muscle cells stacked on top of one another, and they're connected up to produce rings, basically, so that when the smooth muscle cells contract, they can cause the constriction of the blood vessel and thereby control the size of the lumen, basically. Okay, then outside of this layer of smooth muscle cells, you have another one of these elastic layers. Okay, and this is called the external elastic lamina. Okay, so here now is the external elastic lamina. Okay, so this is the external, and then it's elastic lamina, just like the internal elastic lamina. This will consist of elastic connective tissue. Okay, now, the smooth muscle cells, which I didn't actually label up at the time, we'll just call those vascular smooth muscle cells, VSMCs here. Okay, the vascular smooth muscle cells and the external elastic lamina together, these two layers have a name as well. Okay, a name that means the collective two layers together. Okay, and the name for the smooth muscle cells along with the external elastic lamina is tunica again, and then it's tunica media. Okay, so tunica means layer, media then means the middle layer. Okay, so this is the middle layer of the arterial wall. Okay, then surrounding the external elastic lamina, you then have another connective tissue layer. Okay, and this is again non-elastic connective tissue here, shown in yellow. Okay, and this final connective tissue layer outside of the external elastic lamina, this is now called tunica adventitia. Okay, so this layer is called tunica, again meaning layer, and then adventitia. Okay, right, so that now is then the structure of the arterial wall, basically, and I'll just mark the edge of the tunica adventitia out here in yellow. Okay, so that is our artery structure, basically. Okay, so now what we can talk about is the pathogenesis of atherosclerosis. What's actually going to happen in atherosclerosis, and we know what we're now talking about with regards to the structure of an artery. Okay, right, so basically in atherosclerosis you're going to get these huge structures being formed in the subendothelial space that are going to push the endothelium inwards. Okay, and these are called atherosclerotic plaques. Okay, so let's now talk about what actually triggers the formation of these things called atherosclerotic plaques, and then we'll see what they're actually made up of. Okay, so basically, initially, the thing that actually causes the beginning of the atherosclerosis pathogenesis is damage to the endothelial cells in some way. Okay, so the endothelial cells misbehave hugely in atherosclerosis, and I'm sorry, I've missed off the A there, I'll start again. Damage to the endothelial cells. Okay, so the endothelial cells are going to hugely misbehave in atherosclerosis. Okay, and they're going to start allowing LDL molecules in the blood to move across them into the subendothelial space, and they're also going to start recruiting inflammatory cells such as macrophages into the subendothelial space. Okay, and it's believed that what triggers the endothelial cells to behave in such a way is damage to the endothelium. So you somehow damage the endothelial cells. So how could you damage the endothelial cells? Well, for instance, examples of what could lead to damage to the endothelial cells are if you have poorly controlled hypertension. Okay, so too high blood pressure, then basically the too high blood pressure is going to be continuously compressing the endothelial cells, crushing the endothelial cells basically. So that's one of the things that could cause damage to the endothelial cells. Another thing is if you've got nasty chemicals in your blood, okay, such as many of the chemicals in cigarette smoke. Okay, those can damage endothelial cells. So if you smoke, many of the nasty chemicals will end up in your blood, okay, and they can damage endothelial cells of your arteries. Okay, right. So whatever the cause is, what seems to happen is these endothelial cells start misbehaving, basically, and they start recruiting things into this subendothelial connective tissue here. Okay, so let me just draw a bigger picture of what we're looking at here. So we don't actually need all of these outer layers initially. Okay, all we need is the endothelial cells here. Okay, so these can represent our endothelial cells. So here are their nuclei, and they'll be sitting on the basement membrane. And then we need the subendothelial connective tissue, which is this orange layer underneath the basement membrane here. Okay, right. So following damage to the endothelial cells, what now starts happening is these endothelial cells start basically an inflammatory response. Okay, they start recruiting white blood cells, leukocytes, from the blood into this subendothelial connective tissue space. Okay, now it's not usually arteries, massive great arteries like this, that initiate inflammatory responses. Okay, usually inflammatory responses occur in tiny little capillaries and post-capillary venules. There the endothelial cells start recruiting leukocytes. Endothelial cells of massive great arteries don't usually initiate inflammatory responses. They don't usually start recruiting leukocytes into the subendothelial space, but something about the damage that is inflicted to these endothelial cells starts causing them to recruit leukocytes from the blood into the subendothelial space. Okay, so one of the key types of leukocytes that you end up recruiting into the subendothelial space is macrophages, which are a great blob of a cell, like so. Okay, now macrophages don't circulate in the blood. Instead, a precursor to macrophages circulates in the blood, okay, called a monocyte. So this is representing the monocyte, which circulates in the blood here. Okay, and I'll just colour in this monocyte here in yellow. And then the monocyte can then be recruited as a site of inflammation, and then it can differentiate into this bigger structure called a macrophage. Okay, so these damaged endothelial cells start binding to monocytes in the blood and bringing them into the subendothelial space. So they bring them between the gaps between neighbouring endothelial cells. So if we have our next endothelial cell here, okay, the endothelial cells are going to start helping the monocytes to get through those gaps and come into the subendothelial connective tissue, where the monocytes will differentiate into macrophages. Okay. In addition, what starts happening is the endothelial cells start allowing LDL molecules in the blood to leave the blood and come into the subendothelial connective tissue, and this is why having elevated LDL means that this process goes much faster, because if you have elevated LDL, you get much more LDL coming into the subendothelial space. Okay, so now what's going to happen then is LDL particles, which we'll just represent like so. Okay, and of course this picture is out of scale. An LDL particle would be tiny compared to a monocyte. Okay, but so that you can actually see the LDL particle, I've drawn it like so. Okay, so the LDL particles are also going to start coming into the subendothelial space because of the misbehavior of these endothelial cells here. Okay, right, so you're bringing in macrophages and LDL particles into the subendothelial space. What happens now then? Okay, so basically macrophages are these massive cells that are designed to clear infections. Okay, they are recruited to sites where there is trouble, where there is some pathogen running amok, basically. Okay, so what do macrophages do? Well, they try to kill the pathogen, okay, and one of the key ways that they kill pathogens is by trying to gas them out, basically, okay. So they start producing a bunch of horrible molecules called reactive oxygen species, which basically react with proteins, DNA, lipid molecules, you name it. They're very, very reactive, okay? And the idea is that you can kill the pathogen by poisoning it out in this way. Okay, now the principal reactive oxygen species that macrophages can produce is the molecule superoxide, okay, which is O2 negative minus. O2 minus dot, okay, superoxide here. So let me just talk you through the structure of superoxide. What actually is superoxide? So hopefully you know the structure of a dioxygen molecule. Okay, so dioxygen, normal O2, is two oxygen atoms double bound to each other. Okay, so here is the double bond, and I'll just draw some dot crosses for this. Okay, so each one of these bonds has two electrons in, one from each of the oxygens. So the crosses can represent the electrons from this oxygen here, okay, and the dots will represent the electrons from this oxygen here. Now each oxygen also will have two lone pairs of electrons. Okay, so here are the lone pairs of this red electron, red oxygen, and here are the lone pairs of this blue oxygen here, so we'll just colour them in. So here are the blue oxygen's electrons, okay, here are the red oxygen's electrons, okay. So, that's a normal dioxygen molecule. How do we turn dioxygen into superoxide then? Well, basically what you're going to do is you're going to give this molecule, which is perfectly stable and happy, another electron, and I'll represent this other electron, this bad electron, as this orange electron coming in here. So you're going to force this molecule to accept another electron. That's going to give the whole molecule a negative charge, okay? But where's this electron going to go? Well, basically what ends up happening is you have to break one of these bonds, basically, okay, and then send the electrons back to their oxygens, and then this new electron can be paired with one of the electrons on one of the oxygens. If I show this, you end up with the two oxygens bound to each other by a single covalent bond now, okay, and one of the oxygens ends up with 3 lone pairs of electrons because it accepts this orange electron. So let's say that this is this red oxygen here. So here is red oxygen's own electrons. Okay, then it has got this new electron, this orange electron, which is there. Okay, so it's now got a negative charge over here because it's accepted that new electron. Okay, meanwhile, poor old blue oxygen over here, it is now left with only 2 lone pairs of electrons, and then it's left with a single unpaired electron. Okay, and when you have chemical structures with unpaired electrons in them, those chemical structures are called free radicals. Okay, and they are incredibly reactive. Electrons do not like being on their own, basically. Okay, so this molecule is incredibly reactive. It reacts with anything, basically, just to try and find another electron to pair that oxygen with. Okay, so this is the structure of the superoxide free radical. One of the oxygens has a negative charge, the other electron has that free electron, which is what this dot here represents. Okay, so macrophages start producing superoxide, this free radical molecule, in the hope that it's going to kill any pathogens that happen to be nearby. Okay, now, of course, there are no pathogens nearby in the subendothelial space, so what does the superoxide do? Well, it causes a lot of damage, firstly, but importantly, it actually ends up reacting with these LDL molecules that have also come into the subendothelial space. Okay, and the LDL molecules now change in a very mysterious way that we don't really understand. Okay, so LDL molecules can react with superoxide, and it leads to the LDL molecules having very different properties, but we don't really know what these different properties are. Okay, so I'm now going to represent this new LDL particle that has reacted with the superoxide molecules in vivid pink here. Okay, so this is now our new LDL particle that's reacted with superoxide. Okay, and it is now known as oxidized LDL, or OX-LDL for short. But as I say, we don't really know what oxidized LDL is. Its definition is literally just LDL that has reacted in some way with the superoxide, but we don't know what constituents of the LDL are actually reacting with the superoxide, okay, and how this leads to these oxidized LDL particles having such different properties, because they do now have incredibly different properties. Okay, so far we've had the LDL particles coming in, reacting with superoxide particles that have been released by the macrophages, and we've now got oxidized LDL being produced in the subendothelial space. What now happens is these macrophages can now actually uptake oxidized LDL. Okay, the thoughts are that the macrophages understand that this oxidized LDL is incredibly dangerous, basically. Okay, and they now want to remove it from the subendothelial space. Okay, so they actually have a receptor which is capable of binding to the oxidized LDL and then endocytosing it. Okay, but this is not one of the receptors that we have seen previously. Okay, this is what's known as a scavenger receptor. Okay, but similarly to what we have seen previously, it results in the endocytosis of the oxidized LDL. Okay, now the scavenger receptor seems to be particularly good at endocytosing the oxidized LDL over the normal LDL. Okay, so what starts happening is these macrophages start taking up the oxidized LDL. Okay, and of course you get a huge amount of oxidized LDL potentially produced here, especially if you're hypercholesterolemic. Okay, so these macrophages are going to end up taking in a lot of oxidized LDL. Okay, so they get huge amounts of cholesterol being delivered into them, so much cholesterol that they have nothing to do with it, basically. So they end up storing it in massive, great lipid stores, basically. Okay, so this is now this building lipid store inside this macrophage, which is all of the cholesterol being stored in one area, basically, as cholesterol esters. Okay, and these cells, which were previously macrophages but are now just becoming these great blobs full of cholesterol, these are now known as foam cells. Okay, so these are key to atherosclerosis. You start getting the formation of these foam cells in the subendothelial space. Okay, and they're formed from these macrophages that you are continuously bringing in because of the problems with the endothelium. And these macrophages are absorbing the oxidized LDL, which they are creating. Okay, so this process continues happening as long as the endothelial cell is damaged, and you produce more and more and more foam cells, basically, in this subendothelial space. And these are going to start producing a bulge, basically, in the subendothelial space that will press the endothelium outwards. Okay, and therefore will start to distort the endothelium of the artery. Okay, so we'll come back to this in the next video, so we'll continue the pathogenesis in the next video, where we'll talk about the fact that the foam cells can actually end up dying, releasing cholesterol into the extracellular fluid. The cholesterol can start forming cholesterol crystals, and we'll also talk about the migration of smooth muscle cells to form the fibrous cap of the atherosclerotic plaque. And then we'll move on to, of course, why atherosclerotic plaque formation can lead to cardiovascular disease, so heart attacks and strokes.